Hello everyone, I am Sudipta and I am going to present the paper Investigating Citation Linkage as a Sentence Similarity Measurement Task using Deep Learning. My co-authors are Professor Robert Mercer and Felipe Urra. So this is the overview of my presentation. At first I will start with the citation linkage, then in the following part I will talk about the problem formulation and our contributions, then the word embeddings, the sentence embeddings, and all other models that we have used in our work. Then I will talk about the corpus creation that we have used for our work. In the following section, I will talk about the models that we used for semantic similarity measurement at the sentence level. The performance of these models are explained in the experimental results, and the limitation and the future work is described in the conclusion. So at first I am going to talk about citation linkage. So citation linkage is the task of focusing on the appropriate text span of the reference paper given the citation sentence. So suppose you are reading a paper and this highlighted text are some sentences that is referencing to some other paper. So this is the citation index, and the current citation index system only gives the name of that reference paper and their corresponding page. So if the reader have to understand what it's trying to say, he would have to read the whole paper or the reference paper, but where he should have to focus only on this highlighted text span. So citation linkage does this thing. So now I'm going to talk about how we have formulated our problem and our contributions. So this citation linkage problem has been modeled as a semantic relatedness task. Given a citing sentence, this framework actually pairs the citing sentence with each possible sentence from the reference document and then tries to determine which sentence pair is semantically similar and which one is not. So this citation context span may contain one or multiple sentences. However, for this work, this span has been restricted to only one sentence. So that's why we have modeled this citation linkage task as a semantic relatedness measurement task at the sentence level. So this semantic relatedness task is actually formulated as a two-class classification which operates on sentence pairs. So given a citing sentence and a candidate reference sentence, it gives 0 or 1. So 1 means the sentences are semantically similar. So that is the focused line given the citing sentence. So there are three major contributions of our work. First, we built a framework that determines the appropriate cited sentence from a cited paper given a citation sentence. The second thing is that we built a synthetic corpus for citation linkage task with more than 60,000 sentence pairs from 3 different biomedical subdomains. And then we developed a method for cleaning and pre-processing the sentences from different biomedical domains. So now I talk about the background strategy. So we have used different word embedding, one word embedding, unsupervised sentence embedding for the annotated sentence pair creation, different attention mechanisms. So for the word embedding, we used fast text because it has the ability to generate the word embeddings for the out of the vocabulary words using the subword information, and which is very essential for chemical names. Then we used the Sent2Vec for which is an unsupervised sentence embedding model for the annotated sentence pair creation. So in the core, it runs actually a continuous bag of words, and it just makes the average of the words for a containing for a sentence to generate the final sentence vector. Then two different attention mechanisms for our work, and I would like to talk about two prominent works for the citation linkage for Hongo and Mercer in 2007. So they conducted their experiment on biomedical domain, and they built a small corpus for that work, which is actually annotated by expert domain. This corpus contains 3,857 sentence pairs with 23 citation sentences and 3,857 candidate cited sentences. The different machine learning models were used in this task. However, their accuracy was little. In 2018, Lee et al. Applied the citation linkage task over the natural linguistics research articles. So for their work, they used both traditional and deep learning models. So for the traditional models, they used the IDF and the checkered similarity, and for the deep learning models, they used word to fake, convolutional neural network, and cosine similarity. Now I would talk about the corpus. So we had a small corpus created by Hongbo and Mercer containing 3,857 sentence pairs. However, this dataset was highly imbalanced with only 81 positive samples, and all the remaining sentences are negative sentence pairs. So the problem is, if we train our model with this corpus, then there is a chance that the model would be biased. So that's why we had to create our. So for that reason, we have developed our own synthetic corpus of 68,898 sentence pairs from 3 different biomedical subdomains: cell biology, biochemistry, and chemical biology. So we are calling this corpus synthetic because it was not annotated by humans rather than by the unsupervised sentence embedding model Sentuvec. So Sentuvec takes both the cited and the citing sentences and then generates the sentence vector. Then we measured the cosine similarity between these sentence vectors, and for different cutoffs we tried to figure out which cutoff is best for annotating the corpus. So we figured out that with the cosine cutoff 0.57 we got the best result, and with that cutoff we built a corpus with 45.89% positive samples. For the validation and test purpose, we used the human annotated corpus created by Hongbo. So we used a Sentuvec model for the annotated corpus creation, but at first we had to train our Sentuvec. So for that reason, a set of 28,310 full text articles containing more than 4 million sentences from different biomedical journals are used to train that Sentuvec. So Sentuvec was trained with multiple parameters and multiple times. Then the best model was chosen against a validation set, which is a portion of the human annotated dataset from Hongbo and Mercer's work. So for the annotated corpus creation, we had to, that means which contains the sentence pairs, we chose 112 papers from this 28,310 articles randomly. And then for the citation sentences, we chose 2,289 articles that cite at least one of these reference articles manually from the web. So now I'm going to talk about the annotated sentence pair creation. So we had 28,310 research papers from PubMed, from which we chose randomly 112 reference papers, and we collected 2,289 citing papers, which cites at least one of these 112 papers. Then we collected only the citing lines from these citing papers and made the sentence pairs with their corresponding reference papers. So thus we got 475,807 sentence pairs, and we fed them to the pre-trained Sentuvec and calculated the cosine similarity between them. So if the cosine similarity is greater than 0.57, then we annotate those sentence pairs as positive sentence pairs, that means the value 1, and the remainings were annotated with value 0. So if we train our model with all the data, then still the model would be biased because the ratio is not good enough. So for that reason, we chose 37,274 negative sentence pairs using a negative selection algorithm. So this algorithm works like this. So for each and every citing sentence, if there are n number of positive samples, then we choose n negative samples randomly containing the same citing sentence. And if there are no positive samples found, then we choose 5 negative samples randomly with that corresponding citing sentence. And thus we created our corpus with 68,898 sentences. So for the validation set and the test set, we used Hospice and Mercer's work. So for the validation set, we took 800 sentence pairs, among which 780 negative samples were randomly chosen from the negative portion, and 20 positive samples were randomly chosen in the very same way. For the test purpose, the remaining 3,057 samples with 61 positive samples were used. So now I'm going to talk about the data cleaning process because this data are from multiple sources. So for the cleaning of the data, at first we had to delete the unnecessary symbols, then we capture different patterns, and finally the symbols and their comments. So the symbols in the very first row, they are not necessary by means of the semantic, so we just deleted those symbols, and the symbols in the second row comes only with the equations, so we just deleted the whole sentence. Then we use the regex in the very first row to capture the equations and replace them with a tag, and we used the regex on the second row to capture only the numbers, and we use the regex on the third row to capture the numbers which comes with a chemical name, and we preserved those numbers as they were. Then we captured the citation indexing systems and got rid of them and replaced the Greek letters with their corresponding word name. So there are the list of the symbols and their corresponding replacements. So the odd columns here are showing the original symbols that we got in the papers, and the even columns are their corresponding replacements. So now I am going to talk about the models that we have used for measuring the semantic similarity between the sentences. So we have actually used variants of Infersent Architecture. These variants are BiLSTM with the max pooling, BiLSTM with inner attention, BiLSTM with hierarchical attention, and the Hierarchical ConvNet. And finally, bootstrap approach is applied for all of these four models to improve the performance. So this is the core of the Infersent Architecture. This is a Siamese architecture. So both the encoder for the citing and the cited sentence are the identical one by means of both architecture design and hyperparameter settings. So both the citing and the cited sentences are fed to the encoder portion and generates one sentence representation. Then these representations are concatenated, then subtracted and pointwise multiplied, and finally all these three features are concatenated again to generate the final feature representation, which is fed to the following fully connected layer, and finally a 2A softmax is used for the classification task. So I'm going to talk about the encoder architectures right now. So the first one is the BiLSTM with the max pooling. So in this case, the words are fed to the LSTM, and each time step we get some feature representations. So max pooling is applied horizontally over them to generate the final sentence embedding. This thing is done for both the cited and the citing sentence. Then we applied two attention mechanisms, the inner and the hierarchical attention mechanism. So in case of the inner attention mechanism, we also use the BiLSTM and then applied the mean pooling over this value, and then applied the attention mechanism to generate the finally sentence representation. In case of the hierarchical attention, this hierarchical attention was first introduced only for the document classification. It is called hierarchical because at first it attends the words to generate the sentences, and then finally attends the sentences to generate the vector representation of the document. But we used only the first portion. In both of these cases, we used 4 heads, so there would be 4 representation for each head, and then they are concatenated to generate the final sentence representation. In case of the hierarchical ConvNet, there are 4 convolutions operations are applied, and with the features from each convolution, max pooling is applied over them to generate the feature representation of that layer, and all of these layer features are concatenated to generate the final sentence representation. Then we applied bootstrap approach. So in that case, the whole corpus is split into 3 different parts. Then at first the model is trained with the synthetic corpus and is used to annotate the data at the partition. Then this model is trained with this first annotated portion only and used to annotate the second portion. Finally, these annotation portion of partition 1 and 2 are used to train the model again and annotate partition 3. Finally, all this annotated data is again used to train the model, and it is tested against the human annotated data. So now I will talk about the hyperparameter settings and the result. So we trained our fast text with different hyperparameter settings, and the best possible hyperparameters we found out are reported on the rightmost column. So for the scientific, you also get the best parameter settings on the rightmost column. So for the Infersent, we started learning with a learning rate of 0.1, then we decreased it if the validation set accuracy decreases by dividing it by 5, and then we did this thing till this learning rate reaches 10 to the power minus 5. The batch size was 50 and the hidden real representation was 512. As an optimizer, we used stochastic gradient descent. So right now I'm presenting the results that we got for all the models, and the metrics that we use to analyze our model's performance are basically the Matthews coefficient correlation, accuracy in percentage, and balanced accuracy in percentage. The reason for taking these three matrices with more importance is because the dataset is highly imbalanced, so the F1 score is going to be low always. However, among these models, the best result was found for the Infersent with hierarchical attention, both in case of the standalone and the bootstrapped one. And even we can see that the Hospice's and Liettel's models performance improved when they are trained on our synthetic corpus, so it shows the quality of our synthetic corpus. So I want to end my presentation with the contributions again. So our contribution is building a framework for the citation linkage task, building a synthetic corpus for training the models, and developing a method for cleaning and pre-processing the sentences from biomedical domains. So still our work has few limitations and which can be improved in the near future. So in our work, the text span was restricted to only a single sentence, so we can try to expand it to multiple contiguous sentences and sub-sentences span. Furthermore, our models can't work with phrases like which is very important for the chemical names like sodium chloride, where there are two words actually but it's representing one identity. So using the tree structures like tree LSTM and tree transformer can solve this problem. Furthermore, the test data that we have used in our study was created only for the method citation sentences, so it would be appropriate to human annotate a test set with a variety of citations types and see how good the proposed method performs on this expanded test set. So, my presentation. Thank you all.